to our presentation based on the following report by Darlin et al. 2003 on the expression of the insulin-responsive glucose transporter GLUT4 in adipocytes is dependent on liver X alpha receptor. The aims of the research paper are to demonstrate that liver X receptors, LXRs, regulate the expression of GLUT4 through direct interaction with a conserved LXR response element in the GLUT4 promoter. The insulin-responsive glucose transporter GLUT4 plays a crucial role in insulin-mediated facilitated glucose uptake into adipose tissue and muscle, and impaired expression of GLUT4 and been linked to obesity and diabetes. Effect of insulin to increase glucose uptake is directly dependent on recruitment of GLUT4.
When blood glucose levels are high, insulin binds to its receptor and triggers a response to several signal transduction pathways. One example shown here is the PI3 kinase pathway. Once insulin is bound, the receptor gains affinity for insulin receptor substrate 1 and phosphorylates it. IRS1 then gains affinity for and binds to PI3 kinase, which in turn phosphorylates PIP2 to PIP3. This is not shown. PDK1 then binds to PIP3 and phosphorylates a range of proteins including ACT. ACT can then associate with GLUT4, a glucose transporter in muscle and fat cells, leading to its translocation on the cell membrane. Glucose can now enter tissue cells. PI3K also activates sterol response element binding protein to assist insulin-mediated gene induction. SERVs are predominantly expressed in the liver and are thought to be involved in regulating the transcription of glycolytic genes for enzymes such as glucokinase marked as GK. We will now look at liver X receptors on GLUT4 expression.
With GLUT4, insulin signals fat and muscle cells to absorb glucose from the blood by binding to the insulin receptor on the surface of fat and muscle cells. When an insulin receptor is activated, it induces the GLUT4 protein to move from reserves held inside the cells. When GLUT4 is at the cell surface, glucose is transported into the cell down its concentration gradient. It is regulated by insulin. Defects in GLUT4 activity have been implicated in some forms of insulin resistance or pre-type 2 diabetes. LXRs are liver X receptors and they are ligand-activated transcription factors of the nuclear receptor superfamily. There are two LXR isoforms termed alpha and beta. LXRs are able to mediate ligand activity transcription of the GLUT4 gene. LXR isoform has a unique role in the adipose expression of GLUT4. Glucose binds and stimulates the transcriptional activity of the liver X receptors. Glucose activates LXR at physiological concentrations expected in the liver and induces expression of LXR target genes, which is in this case GLUT4. Last 3T3-Li cells were cultured 2 days post-confluence and allowed them to differentiate into adipocytes in order to examine if the introduction of GLUT4 and LXRA alpha overlaps during differentiation. When the differential process occurred, samples were taken each day from day 1 up until the 7th day. Figure 1a shows receptor PPAR gamma transcript was induced remarkably from day 2 to day 3 differentiation, with an overlapping induction of a set of characterized PPAR gamma target genes. On the other hand, GLUT4 and LXR alpha transcripts were induced 24 hours escorted by induction of STREB1, which is the LXR gene target. There is a region where the DNA sequence is similar in both human and mice cells. This is important for drug development in diabetes in humans. Now let's look at the findings. The area circled red shows the transinfection with a human GLUT4 promoter. To examine the validity of the particular LXRE found in the human GLUT4 promoter, they cloned this promoter and inserted it into the PGL3 basic reporter vector. Transient transinfection of this luciferase reporter into the cells was in combination with RXR and LXR expression vectors and treatment. The reported activity was induced. A maximum 12-fold increase in reporter activity was observed following the addition of both receptor expression vectors and ligands. This explains that there is a link or connection between this particular LXRE and GLUT4 transcription.
Here the findings are depicted in a graphical representation of real-time PCR analysis. In Figure 6a, on injection of insulin, wild-type mice did not show altered GLUT4 expression. Figure 6d shows treatment with LXR activator had no effect on GLUT4 expression on LXR alpha or beta isoforms in mice. In Figure 6e, expression of basal GLUT4 in LXR alpha double-deleted mice were lower than other groups. Moreover, there was no increase of GLUT4 expression after insulin treatment. Darlin and his colleagues concluded that LXR alpha isoform is specifically important in insulin-regulated expression of GLUT4 in epididymal white adipose tissue cells of male mice.
Both reduced amount of GLUT4 and recruitment of GLUT4 is strongly associated with obesity and type 2 diabetes. GLUT4 expression in tissues is activated by LXRs. LXR alpha plays a unique role in the basal regulation of adipose GLUT4 expression. It is unclear whether this regulation mechanism is mediated exclusively through LXRs or whether PPAR also regulates GLUT4 expression. Experimental results indicate that LXR alpha expression is important for expression for GLUT4, but it is not totally dependent on LXR alpha. This was found for both human and mice adipose differentiation. Studies suggest that there are distinct differences between LXR alpha and LXR beta isoforms regarding GLUT4 expression. LXR ligand was fed to wild-type mice for 7 days. The control-fed animals' GLUT4 expression in epididymal or subcutaneous white adipose tissue were the same. Results imply that there must be a mechanism which stops prolonged induction of GLUT4 via activation of LXR. Insulin resistance is clearly linked to adipose expression of GLUT4 as its expression was reduced in rodents that had insulin deficient adipose tissue of type 2 diabetics and obese people. The report concluded that activation of LXR alpha isoform in adipose tissue could be of future clinical significance in management of type 2 diabetes.
Thank you for listening to our presentation.